Okay, our last speaker is Wehman, and she's going to tell us about what happens to the second polar body after extrusion.
Thanks. We just have to get back to the first slide would be great. There we go. So thank you to the organizers and also to the organizing committee for selecting my postdoc's abstract for a talk. And today I'm going to tell you about his work on a new project looking at what happens to the polar body after its release at the end of the germline. And this is based on my lab's general interest in released organelles and also in extracellular vesicles. And the polar body is a great example of a released organelle. So as we heard at the end of meiosis, so you have your germ cells. Unfortunately, the mouse isn't working again. Is there any hope? Maybe I'll just switch to the pointer. So we have the backup pointer here. So you have your germ cells which are proceeding in mitosis, they enter meiosis, they freeze for a while until they get activated and fertilized, and then you have an oocyte who has a 4N quantity of DNA. Are we fighting over the mouse now? And it needs to get rid of this extra 3 copies of DNA so that there's only one maternal copy that gets contributed to the embryo. And this is released through 2 extremely asymmetric cytokineses, where 2 copies get thrown out in the first polar body and 1 copy gets thrown out in the second polar body. And this is, of course, important to avoid aneuploidy. So this is what polar body extrusion looks like. It's a pretty well-studied process. So we're zooming in on just this little portion here, and you can see this asymmetric spindle setting up right at the surface of the cell to extrude out the polar body so that it's sitting outside. And you can actually see it by light microscopy because it's pretty big, about 2 microns. So this is something that, of course, not only happens in C. elegans, but also happens in humans as well. And it's been used for genetic testing because this is a way to check for maternal alleles. And it's sort of an indirect way because you're basically, by process of elimination, following which allele was not inherited by the embryo. So this is, of course, much better in a way than testing the more typical genetic testing method that was used before, which is to use a blastomere from the embryo instead of just taking a little polar body off the top. And it's also something that can be done a lot earlier than removing the blastomere, since you can actually do this within a day. But the only problem with this is that actually there's very, very little known about what happens to the polar body after its release. And this is true in C. elegans and also in virtually every other system. And so it's not clear whether the polar body actually gives any instructive cues during development. So we wanted to look in C. elegans at what happens to the polar bodies, and just going to break it up into the first and second polar body because they have fairly different fates. So the first polar body is actually released prior to the formation of the eggshell, and so it actually gets trapped in layers of the eggshell and remains in the eggshell, whereas the second polar body is released after the eggshell is already formed, and so it sits right next to the embryo. And so you can see this quite nicely. Here's the first polar body, which is sitting next to the 1-cell embryo. And if you look at an embryo at any other stage, you can still find the first polar body sitting in the eggshell, and it stays there until hatching. So the fate of the first polar body is not especially exciting. So we'll jump straight to the second polar body. And so the nice thing about this movie is that you can actually watch both polar bodies at the same time. So this is the first polar body again, and this is the second polar body. And you'll see that the first polar body does absolutely nothing, while the second polar body gets internalized into the cell. So it floats around on top, it gets swept in on the furrow, and then pops into the cell. And we'll see if we can play that again. Nope. Let's try. Yeah. So just showing that in still images, it's sitting normally on the anterior side of the embryo. And then when the embryo starts dividing, when the anterior cell starts dividing, you see it riding the furrow in between the cells, but it doesn't actually internalize until after this stage. And this is very dramatic if you keep track of what stage this happens. So if we look at the timing of polar body internalization, there's this dramatic pause in between two waves. And this pause is set off with the division of the AB cell. So when this anterior cell divides, there's a time for about 5 minutes when basically you don't see any internalization of the polar body. So this basically says either the 2-cell embryo internalizes it by AB, or the 4-cell embryo internalizes it either by ABA or ABP. So it's completely random which cell between the two internalizes it. So we wanted to try to figure out how this internalization was happening. And now I've switched my markers a little bit. So before you were looking at histones marking the DNA in the polar body, but now we're looking at NMY2, which is in the actin-myosin ring that closes down when the polar body is released. So you're just looking at a little yellow ring on an empty polar body. But I think you'll very nicely see the internalization event looking at the actin. So it gets swept in, it's sitting there. And then you see this big wave of actin internalizing it. And we'll see if I'm more successful. Yeah, there we go. So it's quite obvious if you follow the blue, you can see it really just wrapping around the polar body. So it looked like this was going to be an actin-dependent event. And if you look at the timing of when the actin happens, as you would imagine, it happens right before internalization. So we assumed that this would probably be phagocytosis, and so we did a little candidate RNAi screen looking for genes that were involved in the internalization of the second polar body, and we mostly focused on genes that were involved in phagocytosis in cell death, cell corpse clearance, for instance, or in vesicular trafficking. And to do the screen, we were looking at one of our ZF1 reporter strains. So I'll just explain to you quickly, if you haven't heard from the Nance lab at some point, about the ZF1 reporters. So this is a trick that's based on work out of Geraldine Sodeau's lab, where they're looking at this zinc finger 1 domain of pi1, which targets proteins for progressive degradation, where it starts getting degraded in the anterior of the embryo, and eventually basically everything but the germ cell goes dark. And released vesicles are protected from ZF1 degradation, and this is true of released organelles also. And they also maintain their ZF1 reporters after they're internalized. So this is what this looks like. So initially at a 2-cell stage, you have it expressed everywhere, including in the polar body. You start getting degradation in the anterior cells, and then the anterior cells have all nicely degraded, leaving your marked polar body right there in its vesicle. So we started looking at whether or not various genes were involved in internalization. And for example, the classical phagocytic pathway with SED1, SED2, and SED10 were all involved, where instead of it being internalized into the embryo, you see it just sitting at some cell-cell contact. So this suggests that's what happens to the second polar body in contrast to the first polar body, is that it undergoes phagocytosis, and that this phagocytosis is based on normal ligand receptor signaling that's leading to RAB-mediated actin polymerization for its engulfment. And this is, as far as we can tell, very similar to the way that cell corpses are being engulfed. So then we started looking at some of the vesicular trafficking hits that we got, and one of the genes that we found was RAB-5 and VPS-34. So this is small GTPase, mostly connected to early endosomes, and VPS-34 is the PI3 kinase that's also tied to early endosomes. And you can see that the polar body just sits on the surface of the embryo and doesn't get internalized in these mutants. And what was dramatic to us was that we actually saw that these proteins were required for the trafficking of the phagocytic receptors. So SED1 is a receptor, and it normally gets trafficked to the plasma membrane. And when you look at RAB-5 or VPS-34 mutants, you don't find it localizing to the plasma membrane any longer. And what we also noticed very quickly was that SED1 was really enriched on the second polar body. And you can see this before the internalization even happens, that there are these clusters of SED1 that appear on the polar body prior to its internalization. So this was really interesting to us because it suggests that these proteins are involved in the trafficking of SED1 to the plasma membrane, but also that there is some sort of feedback where the polar body is actually signaling for SED1 to be trafficked to the plasma membrane. And at this point, we have no idea what the positive feedback loop would be, if it's SED1 signaling itself or something else. But we think this is a really interesting topic for looking at in the future. So it gets internalized, but what happens after internalization? So here we have a rather longer movie. Again, we have the first polar body that's not going to do anything, and the second polar body that you should keep your eye on. And looking at its internalization, it'll float around the cell for quite a while, and then we started to see this sort of budding behavior where it'll split into smaller pieces, and these smaller pieces disappear pretty rapidly, but it actually takes several hours, typically around 2 hours, for the polar body to completely disappear, and that's looking at the histone markers. So, as I mentioned, most of the polar bodies split a few times before they disappear, and that most of these are disappearing within about 2 hours. So we think what's happening to the polar body is that it's being internalized. It undergoes this sort of budding, although proteins that regulate the budding of sort of double membrane structures like this are not really well understood, and that this is then leading to degradation. And at this point, we haven't looked into the proteins that are responsible for either the splitting of the polar body or for its degradation. But we think we have a hint, at least in terms of the degradation, and this is based on another project that my postdoc has been working on, and that's looking at what happens to the midbody after cell division. And so when the one-cell embryo is dividing to a two-cell embryo, you have the midbody that's clustering this event, and it's also a released organelle, so it ends up sitting outside the embryo in between the cells, and it gets phagocytosed by a very typical SED1, SED2, SED10 sort of pathway. And then what's sort of unexpected is that a number of proteins that were thought to be involved in autophagy, but are instead involved in this sort of non-canonical autophagy mechanism called LC3-associated phagocytosis, cause the midbody phagosome to then mature, acquire LC3-associated proteins on the outer surface, which help with the fusion with the lysosome and eventual degradation of the midbody. So we think it's very likely, since the polar body phagosome will also be a double membrane structure, that it's also probably going to undergo this sort of LC3-associated phagocytosis, but we haven't shown that yet. So what I have shown you is that we've started looking at the polar bodies and what happens to them, that they're capable of sending receptor-mediated signals, and so we think that the polar body is not just a piece of trash, it's actually talking to the embryo, and we think it'll be really interesting to sort of look at some new membrane biology in terms of how these double membrane structures are being processed and degraded. And just to bring this back to the beginning, so the hope was that removing the polar bodies was sort of a non-invasive thing. But if this is a structure that's sending signals, then that suggests that maybe this is not the best of ideas. And there was a clinical trial that's been done looking at whether or not polar body testing is actually causing any risks, but unfortunately the results have not yet been published. But one thing that's sort of interesting is that the polar body actually ends up sitting right next to the first cleavage axis in mammals, and so it's possible this could be sort of the bud scar similar to yeast, or it could be some signal that the released polar body is actually sending. So we're really interested in trying to find out. One of the polar bodies in mammals definitely sticks around for quite a while, but no one's really done a careful analysis looking at whether both of them stay. So we're trying to, we'd love to find some collaborators who are interested in helping us with this question, and we're going to keep focusing on C. elegans to look at what happens to what role the polar body could have in signaling during development. So as I mentioned, this was my postdoc's work, Ahmed Fazeli, and he has been helped by a number of undergraduate and medical students who've been in the lab. We have collaborators at the University of Würzburg who are EM specialists who've been very helpful also. And of course, we have to thank the CDC for strains and members of the community as well for your reagents. And I should say that we're also from a little bit further away. So this is Würzburg in Germany, and we have some open positions if anyone is interested to living in lovely Europe. We're not going to elect Trump, so it should be nicer.
Hi, what's your thought on what's on the membrane or not on the membrane of the second polar body that make it different from the rest of the other cells that induce the other cells to engulf it?
We're not sure, since most of the proteins that are involved in the engulfment of cell corpses are involved. We are going to assume that it's something like phosphatidylserine would be a possible thing, but we haven't actually done the staining to see what the ligands would be.
What is the consequence of just a big body ablation?
What's the consequence of what?
Laser ablation.
So laser ablation of the polar body, I don't know if anyone has done it. Laser ablation of the mid-body, this has been published by Christian Pohl's lab, and they saw effects on dorsal ventral patterning in that case. But of course, when you ablate the structure, you're very likely damaging the neighboring areas, so it's kind of a crude way. So we could try dissecting them off and seeing if we could have living embryos, but this is not an experiment I necessarily want to do.
Hey, so I was trying to remember, are there any similarities between this process and the removal of the paternal mitochondria? Are there some of those players are they shared?
So the paternal mitochondria is degraded by autophagy, so typical normal double membrane wrapping around a cytoplasmic structure. So you actually end up with a quadruple membrane structure where you have the two mitochondrial membranes plus the two autophagy membranes.
But no blebbing or anything like that, nothing like that was occurring.
Have people seen them? I don't think I've seen people do any movies of them, so I haven't seen any data.
Hi, just a quick question/comment. Have you looked at mutants that don't do the second polar body exclusion? There was some minority mutants that don't.
No, we haven't played around with any polar body extrusion mutants at this point.
So just anecdotally, they do have some asymmetric cell division defect. But I don't know much more than just that.
Okay, thanks.
I was wondering whether you looked at the machinery that is used usually if you have envelope viruses infecting cells.
Yeah, we haven't looked at those yet. Great, thank you.
Okay, let's thank all the speakers again. We might be able to convince some of them to stay and talk to you if you want, and otherwise, have a good night.